Good morning everyone. So in last class we discussed about quadrant. We understood quadrant, right? Today again we will discuss some more examples because in last class this topic was not completed perfectly. Okay. So today again we will understand this quadrant. We discussed that total we can break 360° in 4 part, 0 to 90, then 90 to 180, then 180 to 270 and 270 to 360. Okay. We discuss in first quadrant which functions are positive. All functions are positive here. Okay. Then in second quadrant which functions are positive? In second quadrant sine theta and cosec theta positive, other are negative. Okay. In third quadrant tan theta and cot theta are positive, other are negative. Okay. And in fourth quadrant cos theta and sec theta positive, other are negative. We understood in last class. Okay. Now important point that we have to understand today, what is it? Today we will discuss that any large angle more than 90°, we can break that angle into smaller angle, acute angle like 0 to 90. Suppose 300° yesterday we discussed. So this 300° we can break into angle which is between 0 to 90. How to break? Understand. If you are writing any angle by using this horizontal line, if you are writing any angle by using horizontal line, then function does not change. What is the meaning? I will explain. First write this point. Horizontal line means function does not change. Function does not change. Okay. And if you are writing any angle, this is important and little bit tough, so all of you listen properly. Okay. So if we are writing any angle by using horizontal line, then that function does not change. What is the meaning? Suppose a angle is there 210, sine 210. Okay. Now 210 we can break. First find that where 210 is, in which quadrant it is. So this is in third quadrant. Now this angle we can write by using horizontal line, otherwise by using vertical line. Okay. By using horizontal line, how we will write it? This is in third quadrant. So 210 equal to 180 plus 30. If we add 30 in 180, then what we will get? 210. So we can write it sine 180 plus 30°. Agree or not? So we use horizontal line. Now if we are using horizontal line, then function does not change. Very, very important. This point is very, very important. Okay. Very, very important. Next, what to do next? So what I said, 180, if 180 is there means horizontal line, and if horizontal line is there, then function does not change, means sign will remain sign. Okay. Now first check that sine 210 is positive or negative. So it is in third quadrant, and you know in third quadrant sine theta negative because tan theta cot theta positive, other are negative. So this is negative. This sine 210 negative. So first use sine. So sine is negative. Okay. Negative. Then what to write? Sine function will remain as it is. Okay. And this angle as it is 30°. And you know sine 30 is how much? 1/2. So this sine 210 value equal to minus 1/2. All of you understood? Okay. Suppose if we use vertical line, then vertical line means vertical line means function will change. Vertical line means function will change. Function will change. Function will change. Sin theta will change. To change into which function? This one you have to remember. So sin theta changes into cos theta. Sin theta changes into cos theta. Cos theta changes into sin theta. Cos theta changes into sin theta. Sin theta changes into cos theta. Okay. Suppose here sin 210, then this sin theta will change into cos theta. If here cos 210, then cos theta will change into sin theta. Understood? So if we use vertical line, then sin theta will change into cos theta will change into sin theta. Tan theta will change into cot theta, and cot theta will change into tan theta. Understood? Cosec theta change into sec theta, and sec theta change into cosec theta. If tan 210 is there, then tan 210 convert into cot. Cot 210 is there, then cot theta will convert into tan theta. Understood? So if we are writing any angle with the help of vertical line, then function will change. And how they will change? Sin will change into cos, cos will change into sin, tan into cot, cot into tan, and cosec into sec, and sec into cosec. Is it clear? Okay. Now next example. Same angle, sin 210. Okay. This angle only we have to write. Sin 210. Now we are writing this sin 210 by using vertical line. Vertical line is here. Okay. Sin 210 means it is in third quadrant. So if we subtract 60 degree from 270, then what we will get? 210. So 270 minus 60 equal to 210. So we can write this sin 210. Sin 270 minus 60 degree. Sin 270 minus 60 degree. Is it clear? Sin 270 minus 60 is sin 210. Okay, so we use vertical line. If we use vertical line, then sin function will convert into cos function. First point. Second point, here you have to check that sin 210 is positive or negative. So sin 210 is negative because it is in third quadrant. So here negative sign. Understood? Okay. And sin will convert into cos. 270 meaning is what? That sin will convert into cos. And finally this angle is 60. So it is 60. Is it clear? Understood? So sin 210 is minus cos 60, and you know cos 60 value is 1 by 2. Cos 60 value 1 by 2. So sin 210 minus 1 by 2. Value will not change. Here you can see sin 210 minus 1 by 2. Here sin 210 minus. Here also sin 210 minus 1 by 2. So value is not changing. Method can be different. We can solve equation by so many methods, but final answer must be same. So final answer is minus 1 by 2. Understood or not? Okay, one more example. Suppose we have to write sin 2 to 5 degree. So can you calculate sin 2 to 5 degree value? So sin 2 to 5 is sin 180 plus 45. Yes, it is correct. Okay. So 180 plus 45, which line we are using? Horizontal line. And horizontal line means function does not change. Sign will remain sign. Okay. Now check it is positive or negative. So 2 to 5 means third quadrant. It is negative. So we will use here minus sign. Okay. Then 45 as it is, and function does not change. So sin 45. Sin 45 value how much? 1 by root 2. Is it clear? Some students are sending, sir, we can write this sine 2 to 5 by using vertical line. Yes, we can write. We can write it sine 270 minus 45. Sine 270 minus 45. Okay. So it is 270 and 270 means function will change. Sine will change into cos. Okay. And check it is positive or negative. This value we have to check. So it is in third quadrant sine theta and in third quadrant negative, to here minus sign. And 270 means function will change. Sine will change into cos and final angle 45. Cos 45 1 by root 2. Now I am giving you some questions, 2-3 questions. These values you have to find out. Sine 330. Okay. Calculate it. Then tan 300. Calculate these values. Okay, so first we will check sine 330. So by using any line, vertical line or horizontal line, you can write this 330. Okay, we can, which line we have to use? You can ask, sir, that which line we have to use, which is simple. So actually horizontal line is simple because here we do not have to change the function. So we can write 330 by using horizontal line. Check it is in which quadrant. So 330 is greater than 270 means it is in fourth quadrant. And you know in fourth quadrant this sine function is negative. So we will use here minus. Okay. And we can break it 360 minus 30. We will get 330. Okay. And final value it is negative. And horizontal line no change. Okay. Sine 30 value 1 by 2. So this is minus 1 by 2. Understood? Now tan 300. Tan 300 we can write it again what I said, that use horizontal line. So tan 360 minus 60. Tan 360 minus 60. Okay. And you know tan theta is negative in fourth quadrant. 300 means fourth quadrant, it is negative. Tan theta negative, so final it will be negative. Okay. And final this angle 60, so it is tan 60. So minus tan 60. Okay. So minus tan 60 value you know root 3, so final answer is minus root 3. So final answer is minus root 3. Okay. Okay friends. So we understood these angles. Now I will ask some questions. These question answer you should give. Okay. Now tell me these values. Sine 90 plus theta is how much? Theta is an angle which is between 0 to 90. Okay. So these value you have to write. Sine 90 plus theta. Number wise you have to send answer. Do not send answer one by one. Send answer. I will write 4, 5. So all answer you have to send together. Sine 90 plus theta. Sine 90 minus theta. Cos 90 plus theta. Cos 90 minus theta. Tan 90 plus theta. Tan 90 minus theta. Sin 180 minus theta. Tan 180 plus theta. Cos 180 plus theta. Send your answers. These all you have to send, and all answer you have to send sequence wise. Okay. First cos theta, second also cos theta hai. One student send cos theta both. Okay, one student send minus cos theta. Yes, correct. Sin 90 plus theta is minus. No, wrong. Minus cos theta is wrong. Why? 90 plus something. 90 plus something means you are in which quadrant? Second quadrant. 90 plus something. And in second quadrant, this sign is positive. Okay. So sign positive, so final here positive. And we are using which line? Vertical. Vertical line means sign will change into cos. So it is cos theta. Okay. Now second, sin 90 minus theta. Again, which line we are using? Vertical line. Sign will change into cos. Now it is positive or negative? 90 minus something. 90 minus something means which quadrant? First. In first quadrant all are positive. So it is positive. So one is friends and both are positive. Yes, that is correct. Both are positive. This is also plus cos theta. This is also plus cos theta. Now this one cos 90 plus theta. Cos 90 plus theta. So in which quadrant this cos theta function is? Second quadrant, because 90 plus something means second quadrant. In second quadrant cos theta is positive or negative? Negative. So we will use here minus sign. Okay. Cos will convert into sine because here which line we are using? Vertical. So it is minus sine theta. Okay. Very good. Most of the students are sending correct answer. Minus sine theta. Yes. Okay. Now cos 90 minus theta. Cos 90 minus theta. So in which quadrant we are? First quadrant. 90 minus something means first quadrant. In first quadrant all are positive. So positive sign. Cos will convert into sine. So it is sine theta. Okay. Now tan 90 plus theta. Tan 90 plus theta. So first of all, which line? Vertical. So tan will convert into cot. We discussed there. You can see here. This tan will convert into cot. Vertical line, tan will convert into cot. Cot will convert into tan. Okay. So here tan will convert into cot. First point. Second, positive or negative? Positive or negative? So here it is in which quadrant? So 90 plus something means second quadrant. In second quadrant tan theta is negative because only sin theta and cos theta positive. So this value is negative. Understood? Now tan 90 minus theta. Again which line? Vertical. Vertical line means it will change. So it will change into cot theta. Now positive or negative? 90 minus something means first quadrant, and first quadrant means all are positive. So it is positive. Sin 180 minus theta. So 180 minus theta. 180 minus theta means which quadrant? Second. 180 is this horizontal line. 180 minus theta means second quadrant. In second quadrant sin theta is positive. Okay. Now which line? Horizontal line. Horizontal line means function does not change. Sign will remain sign only. Okay. Now tan 180 plus theta. Tan 180 plus theta. So again here which line? Horizontal line. Horizontal line means no change. Tan will remain tan only. Now check positive or negative. Positive or negative. So it is positive. Why? Because 180 plus theta means third quadrant, and third quadrant means tan theta and cot theta are positive, other are negative. So it is positive. Cos 180 plus theta. Cos 180 plus theta. Cos 180 plus theta, to which quadrant? Third quadrant. 180 plus something, third quadrant. Third quadrant means cos theta, negative. So here minus sign. Okay. Which line? Horizontal line. Horizontal line means function does not change. Cos will remain cos only. Okay. So we understood all this. Now I can say that you all can find any value, okay, by using these concepts, and correctly you will solve that, I believe, because you all understood. Now I am giving you 3 problems. These are little bit different. So let's see who will correct these. Only those who are minded, they can only solve these questions. Okay. So let's see. What I am writing, only sine minus theta. This sine minus theta value is how much? This one you have to find out. Similar like that, cos minus theta also, and tan minus theta. These 3 values you have to find out. Sine minus theta, cos minus theta, tan minus theta. Okay. So let's see how to solve them. Understand. Sine minus theta. Okay. So first of all, we don't have sine minus theta. How to find? We don't have any concept. But actually, we can write this sine minus theta. How? We can write it like this. Sine 0 minus theta. Agree or not? Can we write sine 0 minus theta? 0 is nothing, na? Right or not? Are you right or not? We can write here 0. 0 means nothing. Okay. Here also we can write it cos 0 minus theta, and here we can write tan 0 minus theta. Agree or not? Agree or not? So now what to do? Why we wrote it 0 minus? We discussed now, this is 0, है ना? This is 90, this is 180, this is 270, and this is 360. So 0 plus theta, understand, 0 plus theta means you are in first quadrant, है ना? First quadrant. 0 plus theta means first quadrant, and 0 minus theta means fourth quadrant. Agree or not? Agree or not? 0 plus theta first quadrant, 0 minus theta fourth quadrant. Agree or not? So we can write this minus theta like this: 0 minus theta, 0 minus theta, here also 0 minus theta. Right? Now tell me that in fourth quadrant sign is positive or negative? Negative. So, and it is which line? 0 we are using, means which line? Horizontal. Horizontal means function does not change. Sine will remain sine, cos will remain cos, tan will remain tan. So this sine theta is negative in fourth quadrant. That's why our final answer is minus sine theta. Is it clear? Many students send minus sine theta. So yes, it is correct. Now cos 0 minus theta means fourth quadrant. Fourth quadrant cos theta positive. That's why here cos theta positive value. Here many students did mistake. It is positive, na? It is not negative. Tan minus theta. So again 0 minus theta fourth quadrant. Fourth quadrant means tan theta negative because cos and sec positive. So here also minus, and 0 means which line? Horizontal. Horizontal line means function does not change. So it is minus tan theta. Clear? So final answer is minus sine theta, cos theta, and minus tan theta. This is correct. Is it clear? Okay, so this topic now completed. It is okay. Now I will start. Okay, new topic I am starting. Straight line. Straight line graph. Okay. So you all know straight line, right? It is not new for you. These all are what? Straight line. This is what? Straight line. Right or not? So here actually straight line we know, but what extra we have to know here? Extra is what? Equation of straight line and its graph according to the equation. Equation means which relate position. Suppose this is a straight line. What is changing here? If we move this side, x will change. If we move this side, y will change. Agree or not? So x and y both coordinate will change. So straight line is the relation between x and y coordinates. Is it clear? Straight line, what is a straight line? It is the relation between x and y coordinates. Okay. So what is the equation of a straight line? Equation of straight line is, this equation you have to remember, it is y equal to mx plus c, y equal to mx plus c. Is it clear? Y equal to mx plus c. This is what? This is equation of a straight line. Okay. Now, in this equation, already I explained you what is a straight line in which two coordinates are related, like x and y or y and z, right? Okay. We are talking about a planar straight line. So here y and x are related. Now y, x, okay, these are the coordinates, but extra is m and c. To understand what is m and what is c, so m is slope of the straight line. Slope of the straight line. What is slope? We will understand it. Okay. And what is c? C is intercept cut by the straight line on y-axis. What is C? Intercept cut by the straight line on y-axis. Okay. Intercept it is. So line will cut the y-axis. How much distance it is cutting? That is the value of C. Understood? Okay. So we understood M and C both. Y and X are the coordinates. Suppose this equation, this graph is given. By using graph we will understand it. Okay. Now what is C here? This is a straight line graph. This straight line is cutting y-axis. How much it is cutting? This much. So this is the value of C. Is it clear? Okay. And suppose this is the straight line. So this straight line is cutting negative y-axis, right or not? This straight line is cutting negative y-axis here. So C value is negative this time. Understood? Okay. Now M is what? M is slope, and slope is measured by tan theta. Slope is measured by tan theta. What is theta here? This theta is angle made by the straight line with plus x direction. What is theta? Theta is angle made by the straight line with plus x direction. So this is plus x direction. Okay, you know, at same time this straight line is making two angle with x axis. One angle is this one, another angle is this one. Agree or not? So which angle we have to take? Which is in anti-clockwise sense. You know, this is clockwise and this is what? Anti-clockwise. So which angle we have to take? Which is in anti-clockwise. Here this is straight line, two angle, one angle this much, another angle this much. Which angle we have to take? Which is in anti-clockwise sense. Is it clear? So this is theta. This is theta, not this one, not this angle. Is it clear? So here M is slope and slope is measured by tan theta. And what is theta here? Theta is angle made by straight line with plus x direction in anti-clockwise sense. In anti-clockwise sense. Understood? Okay. So we discuss equation of straight line. Then here two quantities are new, m and c. What is m? M is slope, which is tan theta. Theta is angle made by the straight line with plus x direction in anti-clockwise sense. Okay. Then we discuss about c. What is c? C is intercept cut by the straight line. Okay. Where at x-axis. So here this is intercept. This is intercept. In this case negative, in this case positive. Is it clear? So only we discuss today these, like, about only this equation. Further we will discuss tomorrow. Tomorrow we will understand that using different graph how to form equation, or by equation how to form graph of different straight lines. These we will discuss tomorrow. Okay friends.
Good morning everyone. Good morning. Is it audible and visible to you? Yesterday we did the degree of carbon, degree of alcohol, halides and degree of amines, right? Clear? So today we are going to start what? Nomenclature. Very simplest topic. I think you have done little bit in your 10th class as well. Yes or no? Yes? So we will understand step by step. First we will see the nomenclature of alkanes, then alkenes, alkynes and having functional groups. Fine bacha? Okay, fine. So let us start nomenclature, right? We will discuss IUPAC nomenclature. Fine? We are discussing IUPAC nomenclature. Okay. So when you want to write the name, IUPAC name of organic compound, which things are required? Basically, totally overall we need 5 things, right? Which 5 things? You have to see. Like first one is, I will say here, secondary. It is secondary prefix, right? After that you will see primary prefix, right? Next thing bache is your word root. It's our word root. And next one I will say it is our primary suffix. You know what is suffix? Suffix means which comes later. Okay. And next is secondary suffix. It is our secondary suffix, right? So these are total 5 things which will include the naming of all of your organic compounds. Right, bacha? Suffix means which comes later, whereas prefix means which comes earlier. Like something which is coming before word root, I am calling it prefix, and that thing which is coming after word root is what? Your suffix. Clear, bacha? So everything is having some role here. So we have to understand the role of all of these things, right? Like in this, you will see the word root is the main part. Word root is the main part. It will tell us the length of main chain. Okay. What is the meaning? What is the purpose of word root? It will tell us the length of parent chain. It will tell us the length of parent chain. Okay. You'll understand when I will take example. Okay. Next thing is primary suffix. Will tell you that our compound is a ane, ene or ine. Means it is having carbon-carbon single bond, double or triple, right? So here you will see it will tell us that our compound is saturated or unsaturated. Okay, ane, ene or ine, right? Parent chain about what? Ane, ene or ine. Clear? Our compound is alkane, alkene or alkyne. Fine, bacha. Secondary suffix will tell us about the function group, will give us about information about the function group. Means it is containing what? Ol or alcohol or acid, whatever. So it will give us information about functional group. Fine. Yesterday we did function group. Like I will say, for alcohol we use ol word, for aldehydes we use al word, for carboxylic acid we use which one? Oic acid. This type of things. So information about the functional groups will come from where? secondary suffix, right? Next thing, what is the meaning, what is the purpose of primary prefix? It will tell us our compound is alicyclic or acyclic. It will tell us our compound is open chain or cyclic. This will give us information about open chain or cyclic. Our compound is cyclic, so I will use word cyclo. If our compound is acyclic, there is no cycle, then I will use what? I will not use anything. Okay, clear? Now next thing is beta secondary prefix. This will give us information about substituents. This will give us information about substituents. Fine, bacha? Clear? Or you can say branch chain, or it will give us the information about branch chain or branching things, right? Clear, Mera? Clear, bacha? So these 5 things are important for us, step by step. I'm saying once again, the main thing which will always be present in our compound is word root, length of parent chain, primary suffix, ane in ine, it is alkane, alkene, alkyne. Second thing is what? You can say secondary suffix, it will tell you the function groups, carboxylic present, ester present, amide, alcohol, amines, what is present, right? Next thing is primary prefix giving us our compound is open chain or closed chain, and this will give you information about the substituents. I am taking one example and using that I will explain, right? Look bache, look carefully. Name I am writing, okay, don't worry about it, you don't try. I am writing the name. I have this compound. First tell me, this compound is cyclic or not? This compound is open chain compound, or better tell me, this is open chain or cyclic? Definitely cyclic compound, right? I am writing the name for this carefully. You don't worry about it. Step by step we'll understand how we are writing the name. I am writing for you. First of all, I am writing for you, right? I am writing the name. It is 3-methyl cyclo hex ane 1-ol. I am giving this example just to tell you the role of every one of these. Clear, bacha? Look at this carefully. Our main chain is hex, 6 carbon chain. So it is our what? Word root. This is my word root, right? It contains ane only. It's having only single bonds, na? It is what? It is your primary suffix. It is containing which function group? OH. So this is the information about function group. What is it? Secondary suffix. Okay? Our compound is cyclic compound. That's why I am using this cyclo word. Which information? It is coming from what? Primary prefix, right? And next, we have a substituent also. Substituent. This is what? This is secondary prefix, right? So you see, everything have its own use. I gave you one example. How to write, we will discuss about all these things. Okay, just step by step. Okay, you'll understand how to find parent chain, how to find it is saturated, unsaturated, everything. Okay, step by step. Okay, bacha, I hope we are clear up to now. Okay, I am going to next page. We'll understand step by step. Fine. Okay, so before starting, there are two major rules which will decide all the nomenclature questions. So please carefully understand this. The first rule is, I will say two main rules. Two major rules you can say. There are many rules in nomenclature, but these two are the major rules. First rule is selection of parent chain. Which chain is our main chain? Selection of parent chain. Which chain I will take as my major parent chain, right? So I am giving a sequence here. That sequence will tell you your main chain should include what first of all? Like I am giving here first of all main functional group. Before selecting the parent chain, you have to take that chain as main chain which have our main functional group, right? After that, the second thing is important for us is double triple bonds. Okay, then third one is what our next one you will say longest chain, longest carbon chain, right? If by chance we will get two, as like two chains are there of same length, then I will take which one? More substituted. If by chance we have two chains with the same length, then I will take it more substituted, means more substituent it should have. Okay, example I will take step by step, everyone. Okay, look at this carefully. First of all, I am taking this part only. I am not taking any pi bond in my question. You have to choose a chain which have longest carbon chain, right? Look at this. I am having this one, right? And here I am going here. I should take straight chain as my main chain, or I should take this as my main chain, or I should take this as my main chain. Tell me. Which chain should be my main chain? Which chain I should take as my main chain? Right, everyone? I should take straight chain, I should take this one or this one. Right? Why I am taking straight as my main chain, bacha? Look at this. If I will take this as my main chain, so how many carbons coming? 1, 2, 3, 4, 5, 6, 7. If I take something else, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So I should take that chain as my main chain which is containing more number of carbons, right? In this there is no pi bond. In this there is no pi bond here. So we will not think about these things, right? I will not think about these things. For here, length is more important. Fine. Next what I am asking, look at this carefully. Carefully give the example. Think about it. I should take which chain as my main chain? Be careful, pi bonds are more important than the length. Pi bonds are more important than length of chain. Pi bonds are more important than length of chain. Be careful about it. I should take straight chain as my main chain, or I should go for L shape. Straight or L? Straight chain or L shape? I am calling this L shape or straight as my main chain. Very good. Downside, L shape, L shape. Why? Very good, very good. This is my main chain. This is our main chain. Very good. Why? Because this chain contains pi bond. This contains pi bond. Maybe straight chain have more carbon atoms, but what is more important for us? The more important is what? Pi bonds. More important is pi bond. Very good. Very good, very good. Clear? I hope you are clear now. Yes, everyone? Good, good, good. Right. Next, I am asking one more question. Look at this. Look, here we have a functional group COOH. Carbon, 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 carbon here, carbon. Right? And here I am going pi bond, or I am doing one thing. I am taking here also the same function group OH, and now here I am taking the longer chain, this one. I should take straight chain as my main chain, or I should take the L shape, straight or L. What is more important for me? Main functional group, right? So I will go like this. I will go like this. Yes, everyone? Okay? Remember one more thing. I will tell one more thing. If in a compound there are two function groups, then only if both are same, like carboxy, then to both are our main function groups. But assume one is carboxylic, one is alcohol. So we have to choose which one is main function group. I will come to that later, don't worry about it. We will come to that point later, right? Now, first of all, simple things we are doing. Now, next thing. One more example and we will move to the next one. Carefully give the answer. Okay, fine. Tell me, I should take straight chain as my main chain or L beta? Straight or L? Straight or L? If you count carbons in the straight chain, how many carbons are there? Count. 1, 2, 3, 4, 5, 6. 4 and 5, yes, 6. In L also how many? 1, 2, 3, 4, 5, 6. But L is also 6. But L also is what? 6 only, right? But L contains more substituent. Length is same, but L contains more substituent. What I mean by this? Understand carefully. Okay, understand this very carefully. If you go here in straight away, if you're going straight away, you're having only one substituent. But in L shape, how many substituents? One substituent here, one substituent here. If length is same, if length of chain is coming same in two cases, then take which one? Take that one which is having more substituent. Clear, bacha? If length is same, I am writing here, in case of same length, right? In case two chains have same length, choose more substituted. As main chain. Fine, bache? Are we clear? I think are we clear? Right, very good. Now next rule I am coming. Next rule. Second rule, bacha. Second rule, the major rule. Second rule is, you can say, numbering of parent chain. Step by step we have to move. Numbering of parent chain. First we have chosen the parent chain. Now we will do its what? Numbering. Clear, bacha? Come to that. Numbering of parent chain. Again, priority wise, if we check, I will go for main functional group. Is important for us. Main functional group. Function groups are most important thing, na? Okay. Next thing, beta. Again, we will choose pi bond importance. Now, from which end we have to do the numbering? We will do the numbering of parent chain from which rule? Main function groups, pi bond, and then very, very important, lowest locant rule. Next is lowest locant rule. But remember, more than these, function group is more important, and then which one? Pi bond. And if every rule become tie, then I will see lowest sum. Then I will see lowest sum of locant rules. But remember, lowest locant rule is more important than which one? Lowest sum. Lowest locant is more important than lowest sum. Clear, bacha? We will see step by step. Okay? I am taking example. Please give me the numbering. Carefully give me numbering. Look at this. First of all, choose the main chain and give numbering, bache. Tell me. Choose the main chain and give numbering. It is main chain. I should start numbering of main chain from where? From this right and left hand. Answer me. Left hand or right hand? Fast, fast. I will start numbering from where? My locand is coming at which location? 2. 1, 2, 3, 4, 5. From which end we should do numbering? Very good. Right. 1, 2, 3, 4, 5, 6. Because it is coming at lowest locand rule. Because it is coming at lowest locand, it is coming 2. From here I am getting 5. Lowest locand rule. Very good. Very right. Fine. Very good. Right? Next one I am saying. This one. Choose this one, beta. I have here C double bond C. I should start numbering of my main chain. This is our main chain, we know very well. This is our main chain. I should do numbering from which end? Left side or right side? I should start numbering from left side or right side? Left. Very good. Why left? Because pi bond is more important. 1, 2, 3, 4, 5, 6. Very right. Why? Tell me why? Because pi bond is more important than which one? Locant rule or any substrate, right? Now, next one. We are moving to this picture. We are having here C. Imagine I have OH here. OH, carbon, here carbon. Tell me now. I should start numbering from which end? Left to right. I should start numbering of this from which end? Left to right. Which end? Left. Very good. Why? What is more important? Answer me. What is more important? Pi bond is more important or function group is more important? Answer me. Pi bond is more important or function group? So I will start from here. 1, 2, 3, 4, 5, 6. Okay, fine. Now one last question, bacha, and then we will move to the nomenclature questions. Okay, give me answer very carefully. Okay, now here we have OH. Okay, I am taking here one more carbon. It's OH here and now here. Now choose which one, left or right. Which end I should opt? From which end I should start numbering? From left hand or right hand? Yes, everyone? See carefully. Function group is where? 1, 2, 3, 4. 1, 2, 3, 4. Function group present at the middle. So you start from any end, function group is same. So definitely I will go from here because I am having benefit of what? Pi bond. Right? If function group is present at where? In the middle, then you can choose, then you can look for what? Pi bond. Pi bond is right side. I hope we are clear here. I hope we are clear about these two rules. These two rules are very important for us. Okay, remaining we'll see step by step. Okay, bacha? Right? Okay. Understand one thing. You know this very well. In your 10th you have done. If you go for word root, okay, I am starting the naming. If you go for word root, if you have only one carbon, if you have only one carbon, what do you call this? Meth, right? If you have two carbons in our main chain, it is what? Eth. If you have three carbons, what do you call it? Prop. If you have four carbons, we have how much? Bute. If you have five, what do you call it? Pent. Similarly, with 6, how much? Hex. Okay? Hex, hept, oct, yes, none, dec. You know that very well. If now imagine these are used as substrates. If beta I am using these as substrate, imagine I am having CH3 as my substrate. If these come as a substituent, side chain, as substituents is come, so how you will name it? Assume my substrate is what? CH3. So I will call it methyl as a substrate. What do you call it? Methyl. if it comes CH2, CH3 as a substrate side chain, it is what? Ethyl. If it comes like CH2, CH2, CH3, instead of just saying ethane, I will call it ethyl. What do you call this? Propyl. Good. In side chain, "-yl" word will come. Alkyl. In side chain, if it is present, call it what? Alkyl. Very good. Now, same thing, bacha, if I say 4, it is what? Butyl in a substrate. I hope you get the point. Very good. So, please follow 2 rules and try to name the alkanes. I am starting with alkane. Very simplest one, alkane. Nothing else. Now, imagine I am saying I am having this one, CCCOH here. Okay, this is my main chain, only one chain present. So I will not start from this carbon. I will not say 1, 2, 3. It is wrong. I will start from that end which is near. It is not necessary to start from here. I will start from here. 1, 2, 3, 4. I hope you get the point now. You will start from that end which is near the function group. You will not start from that carbon which is having function group. Okay? Clear, cha? Very good. Now I am coming to the alkane. Okay, we are doing some examples of alkane now. Okay, bacha? So we are doing questions of alkane. Carefully give the answer. Don't make mistake. The first one is just simple one. Okay, second one I am saying here. Okay, third one I am saying here. This one, step by step we are moving. Okay, fine. Yes, everyone, try the first one. What is the main chain? Only one main chain, only one chain is present, so no doubt you can start from any end. 1, 2, 3, 4, 5. Just simple thing. It is what? Pentane. Pent is word root and it is only single bond. So ane. Pentane. In all the names, it is not necessary that function group will come, cyclo will come. No. Whatever is required, you have to use it. In this only, I am only using what? Word root. This is my word root. And this is my what? This is word root. This is a primary suffix. Nothing else required. Nothing else required. Right? It is not cyclic, so nothing required. No function group, not required. Right? Next one. Name this fast. Now, definitely I will start from here because by the lowest locant rule, substrate is coming at low corner, right? Low position. So what I name this? 2-methyl. 2-methyl, what is it? Pentane. 2-methyl pentane. Okay, 2-methyl pentane. Now see carefully what we have used here. We have used word root. We have used primary suffix. We have used what, beta? Secondary prefix. Other things are not required, so leave it. Whatever things are required, name it. Okay? This is our main chain. This is our what? CH3 substrate. Substrate means methyl. What location? 2 location. 2-methyl pentane. Very good. Great. Now, next one if I go, what is next one? This is next one. Okay? I should start from which end? This end? 1, 2 or 1, 2, 3. I will start from here. Why? Because of lowest locant rule. Very good. Chakri beta, very good. Rockstar doing good. Swati doing good. Everyone is doing great. And everyone is giving answers. It's a very good thing. Great. So, our substrate at which location? 2 location and 4 locations. What I will say? I will say 2, 4. As there are 2 methyls, I am saying dimethyl. 2, 4 dimethyl hexane. 2, 4 dimethyl hexane. Very good. I am saying once again. What is this? This is your secondary prefix. This is your word root. This is your primary suffix. Okay? Whatever we need, you have to use accordingly. Whatever we need, we have to use accordingly. Fine, bacha? Very good. Fine, fine. One more question I am giving here. Answer me. Answer carefully. Right. Right name for this. It is our main chain. Okay. 1, 2, 3, 4, 5, 6, 7. Or you can say same thing. What is the name of this substrate? If you see carefully, how many carbon? 2 carbon in straight chain. 2 carbon in straight chain. It is ethyl. It is ethyl. Its name is ethyl. As a substituent, its name is ethyl, right? Rectify your answer now. Some students are doing great. Look at this. I am calling it 4-ethyl. 4-ethyl, right? How many? 7. Heptane. Heptane. 4-ethyl heptane. 4-ethyl heptane. Very good. Try this one. This time make it right. Don't make mistakes this time. Don't make mistake this time. Give me answer fast. 1, 2, 3, 4, 5, 6, 7, 8. Tell me its name. Exactly same. Substituent is ethyl. CC is a substituent, so it is ethyl. Okay? Right. Name is 3-ethyl. 3-ethyl octane. Very good. 8 carbons in main chain, 3-ethyl octane. What is it? If you see step by step, what is it? Secondary prefix. Word root. What is it? Primary suffix. Okay? Great. Now I am giving a little complicated. Answer this. Carefully give the answer. Look at this. This is our main chain, and this time it have 2 substituents. I will start from here because lowest locant rule. I have 2 substituents. We have 2 substituents. One is methyl, one is ethyl. We have here 2 substituents. One is methyl, one is ethyl. Right. Give the answer. Okay. If you have confusion, I am writing it once again a little bit. It's carbon, right? This is our main chain, okay? This is our main chain. Very good. Now remember, when there are 2 substituents, you have to write them in alphabetical order. When there are 2 substituents, you have to write them in alphabetical order. I am having ethyl, I am having methyl. So E comes first. Ethyl, then methyl, and then the name. Right. Very good. Right. Very good. What is the name for this? Tell me first of all. I will say 4-ethyl. 4-ethyl, 3-methyl. 4-ethyl, 3-methyl. How many better now? Octane. Very good. 4-ethyl, 3-methyl, octane. Right. When there are 2 substituents, you have to write them in alphabetical order. Like you see, beta, there is ethyl is substituent and methyl also what? Substituent. So you have to write ethyl first. You will write ethyl first instead of methyl, because E comes first, then which one? M. Ethyl, E. methyl M in alphabetical order is coming first. That's why you will start with E. Clear? Now same thing I am giving once again. Carefully give the answer. Be careful. Right? Okay, this is our main chain. Okay, numbering I am giving to you. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5. No, this is a rule, right? Now, one substance is ethyl, one substance is methyl. Okay, bacha? I am telling you the name. It's ethyl substituent. Ethyl. What is it? 3 carbons means propyl. 3 means propyl. Now write the name in alphabetical order. If 2 substances are coming, write name in alphabetical order. Out of E and P, who is coming better first? Definitely E coming first, na? So I will say 4 ethyl. 4 ethyl. How much? 5 propyl, right? What is it? How many beta? Nonane. 9 means nonane. It is nonane. Clear, bacha? Are we clear? 4 ethyl, 5 propyl, nonane. Very good, very good. Okay, one last question for today. One last question for today. Tomorrow we'll see questions from PDF, right? Look, bacha, look at this. If you give this question right answer, that means you know the topic. Now 2 methyl groups are present on same carbon, so carefully give the answer. It is our main chain. It is our main chain. 1, 2, 3, 4, 5. How I will name it? Well, very good, very good. 2 dimethyl. If substrates are same, then call it dimethyl. 2 dimethyl pentane. 2 dimethyl pentane. Fine? Very good. I hope we are clear. Okay, bache, so tomorrow we'll do questions from alkane. Okay, and then from alkene alkynes also. Okay, okay, everyone. I hope this was helpful for you. So don't forget to subscribe and like, right? And don't forget to fill the form also, right? In the description. Okay, bacha, bye. See you tomorrow. Tomorrow we'll do questions from alkane alkynes also. Okay, bye.
Good morning. How are you all? Did you read yesterday's class? Whatever the topic I have taught. So yesterday we had completed the prokaryotic cell. I don't want to ask the questions yesterday because I was giving the first lecture for you. So I couldn't interact much with you all. So today let us hope I will be doing the more clarification of your doubts and all. Yesterday we have done with the prokaryotic cell. Is it right? Yes, very good, very good. So prokaryotic cell, I'll be asking some questions, very few questions, okay? Examples of prokaryotic cells which I gave yesterday. It's very good, bacteria, very good, bacteria, correct. Now, I would like to ask you one more question in this and later we'll continue with the topic. So now, I would like to ask you one simple question. Name the structure of bacteria, okay, which gives them the name prokaryotic cell. The main structure of the bacteria which gives the bacteria that it is a prokaryotic cell. What is it? Find out among the given options. Nucleoid, first option. Second option, nucleus. Third option, ribosomes. Fourth option, plasmid. Which one is correct? Yeah, the answer for this is nucleoid. This is the answer for that question. Nucleoid is the nucleus without any nuclear membrane. So that kind of nucleoid it is called as, that is present in the prokaryotic cells. That is why the bacteria got the name prokaryon. That means primitive nucleus, the nucleus which is not bounded by any membrane. Last one more question before going to the actual topic. Does this prokaryotic cells contain any kind of mitochondria, chloroplast, ER? Do they have? Do they have any of these cell organelles? Prokaryotic cells. So all of you are correct. So there are no membrane bound cell organelles. Very good, very good. So now I came to know that you understood yesterday's lesson. Okay. So now let me proceed with the next point in the lesson, same lesson, cell the unit of life. Prokaryotic cell you have learnt already. Now let us see the eukaryotic cell. Before going to that eukaryotic cell, you know some of the features. How to differentiate the prokaryotes from the eukaryotes means, based on whatever the points yesterday I have given you that prokaryotic cell will be having, generally they are absent or some of the structures are present here. In that way you can differentiate the prokaryotic cells from the eukaryotic cells. For example, I am taking nucleoid. Nucleoid is present only in prokaryotes. In eukaryotes, instead of that, nucleus is present. No nucleoid. Okay. Cytoplasm is present in both of them. And motile forms will have the flagella here also. Flagella are present here as well as here. Pili and fimbriae. These are the surface structures which are present only in this. They are not there in that. But cell envelope is there. Cell envelope. This is present here and this is absent here. So these are the main differences between the prokaryotes. Here you are learning just the brief explanation. This is just the introduction or basic information what you are getting. The further details of this topic, when you come here in the offline classes, you will be learning. So I would suggest you just read how much ever I am teaching you in the class. Remaining things, yeah, it's not prokaryotic cell is not completed yesterday only. No, it has so much of information which you will be learning after coming here. Okay, I hope all of you understood this point. This is just the basic information. Okay, so now the prokaryotes and eukaryotic cell differences you know. Now let us consider the eukaryotic cells. Yesterday I told you which are the eukaryotic cells. Can you tell me the examples of eukaryotic cells? Very good. Fungi, animal cells. Very good. So eukaryotic cells are from fungi. They are algae. Then plants and animals. Today our topic is in the plant cell and in the algal cell and in the fungal cell, what is the outermost structure, outermost cell structure? What do they have? You know, in all these three, cell wall is present. The common character which is seen in all these three, algae, fungi and plants, is they contain cell wall. You know that animals, they don't have the cell wall. So here let us see, in these organisms, cell wall is outermost structure and this cell wall is present in them and it is made up of different kinds of materials. Let us see the first, the cell wall structure. Yesterday in the cell envelope you have learnt that the outermost one is the glycocalyx in the prokaryotes. Next, inside one is the cell wall. Inside one, from outer side to inside, this is the plasma membrane. This is what you have learnt yesterday. Am I correct? This is what you have learnt yesterday in the cell envelope of prokaryotic cells. We have got from outside glycocalyx, then cell wall and plasma membrane. Glycocalyx is having the capsule. So now in the plant cell, algal and fungal cell, the outermost one is the cell wall. So by this cell wall materials, we can differentiate these three organisms. Animals anyhow, they don't have the cell wall. So in them, the outermost membrane is the plasma membrane itself. Okay. Now let us see the cell wall structure. All of you can see that green color, generally plants are green in color. So So I have taken the cell wall also with that green color boundary, and this is the cell wall, how it looks under the microscope. This is the enlarged view of the cell wall and a cell. And all of you can see here, this is the nucleus. We are not concentrating in the cell organelles and the cytoplasmic contents. Here we are concentrating on the cell wall. So this green line, can you see these three parts? I hope all of you can see these three parts. Yes, yes, students? Yeah. So here these three are together called as cell wall. So cell wall is made up of, you can see that diagram and you can tell me, cell wall is made up of how many layers? You can see the diagram and you can tell me. Very good, three, very good. So all of you can see that 3 layers are there. The first one is the middle lamellum. Middle lamellum is the first one, very good. From outside to inside we are going again. Okay. The outermost one is the middle lamellum, and the second one is primary wall, and the third one is secondary wall. So when you draw the diagram in the cell wall, the outermost layer which connects the two cells So all of you can see that this is one cell, this is the second cell, the other side. This is the neighboring cell. So 2 neighboring cells I have taken, and one cell and the next plant cell, they are connected by the middle layer, the white line. This is called as middle lamellum. Middle lamellum. This is the first layer. So middle lamellum is the outermost layer of the cell wall, which is made up of this layer. It is made up of calcium pectate. Calcium pectate. It acts like glue. It will be gluing, or it will be connecting, or it will be cementing the 2 layers together. The 2 cells will be connected together because of the middle lamellum, which is made up of calcium pectates. So what is the main material with what the middle lamellum is made up of? Calcium pectate. So this is the cementing layer, and it will be connecting the 2 cells together. Okay, that is about the middle lamellum, which is the outermost one. Then next layer, internal to that we have the primary wall. This yellow color one, this is the primary wall, second one. Primary wall. Always on the inside of the middle lamellum only, the primary wall is formed. Always it is formed on the inside. And primary wall, generally I am taking the plant cell because this is the plant cell I have taken. So in the plant cell, the primary cell wall is mainly made up of cellulose. The materials which are there in the primary cell wall are cellulose, hemicellulose, pectin and proteins. So primary cell wall is made up of 4 materials, mainly cellulose, hemicellulose, pectin and proteins. So this is always formed on the inner side of the middle lamellum. Okay. So primary wall, it is formed on the inside of the middle lamellum, and it is formed as a first layer from the cell wall. You know, middle lamellum is connecting the 2 cells together, but that is not the first formed one. The first formed wall layer, cell wall layer, it is called as the primary wall. From here the cell wall will begin. Though middle lamellum is the outermost one, the first formed wall layer, because middle lamellum, it is not called as middle wall or something. They did not give the name as wall. So they have given simply the primary wall is given the name. So the first formed wall layer, cell wall layer, is the primary wall, and it is formed generally on the inside of the middle lamellum always. And then inside the primary wall, we have one more layer which is called as secondary wall. In the secondary wall, this green one is the secondary one, secondary wall. In this you can see this one is a secondary wall, okay, and this boundary edge, this is the primary wall, this is the primary, whereas this blue one is a secondary, and the gap present between them is the middle lamellum. So secondary wall is formed on the inside or outside of the middle lamellum? Middle lamellum. To the middle lamellum, secondary wall, is it formed on the inside or on the outside? It is formed on the inside. Always the secondary wall is formed on the inside of the middle lamellum, particularly on the inside of the primary wall. Yes, yes, very good. So first middle lamellum, then primary wall, and then secondary wall. Secondary wall is always formed on the inside of the primary wall, and it is also made up of different materials. In this, the materials which are forming the secondary wall will be lignin and suberin. So secondary wall is made up of lignin and suberin. So it is generally formed on the inside of the primary wall, and it is made up of different materials like lignin and suberin. Always on the primary wall, the secondary wall will be deposited, and the secondary wall materials are lignin and suberin. And this primary wall always will be growing until the plant cell reaches maturity. After that, the growth will be stopped for the primary wall, and on the inside of the primary wall, secondary wall will be formed in the plant cells. So this is the structure of the cell wall and the functions of the cell wall. Let us see that functions. I hope all of you understood with what materials they are formed. I am going to the next page, and here you will be answering to my questions. Okay. The functions. Functions of cell wall. First thing, it gives protection. Okay, it will be giving the protection from the external environment, from different organisms, from different animals. The cells are being protected by the cell wall. That is the main function. Next one, it gives definite shape to the cell. So as it is outermost boundary in the plant cell, algal cell and fungal cell, they will have a definite shape because of the cell wall. And sometimes it acts as a barrier for the big macromolecules and all. They cannot enter through the cell wall, big ones. It acts as a barrier. But in general, the soluble substances, all of them, they can cross a cell wall, and it acts as a permeable membrane, either this or this, depending on the molecular size. On molecular size. If it is a very big molecule, it cannot cross the cell wall, so it acts as a barrier. If it is a small molecule and if it is soluble molecule, it can cross the cell wall easily. Then cell wall will be acting as a permeable layer. And remember, cell wall is always dead. It is always dead structure. Okay. So this type of plant cell wall is there with the 3 layers. Middle lamellum, primary wall and secondary wall. Middle lamellum is a cementing layer which glues 2 neighboring cells together, and it is made up of calcium pectate. Whereas primary wall, it is formed the inside of the middle lamellum, and it is made up of calcium pectate. And this primary wall is a first formed cell wall layer always on the inside of the middle lamellum. And inside the primary wall, we have the secondary wall. And secondary wall is the last formed secondary wall is the cell wall layer, and it is always formed on the inside. And inside that, what is present, you will have a doubt. Inside that secondary wall, we have the plasma membrane. And plasma membrane we will see in this one only after this. Secondary wall is made up of which materials? Lignin and suberin. Okay, either lignin or suberin or both of them will be deposited. It depends on the cell type. Very good. Then next question. Very good, very good. Middle lamellum is made up of which materials? Middle lamellum is made up of which materials? Calcium pectate. Very good, very good. Now tell me primary wall materials. Primary wall is made up of which materials? Yeah, cellulose, very good. Hemicellulose, yes. Yes. Pectins and proteins. Yeah, right. Now, the functions of the cell wall. What are the basic functions of the cell wall? I have written here. Okay. Now, I will go for another page, then you can answer. Because I can't ask you the questions after placing that one on the board, no? Yes. Functions. Tell me any one function of the cell wall. Any one function. Yeah, pectin, correct. Okay, protection. Very good. Protection. Yes. Is it a barrier or a permeable membrane? If the molecule is big, it acts as a barrier. Right. It acts as a barrier. For small molecules, soluble substances, it acts as, for the big molecules, barrier. For the small molecules, soluble substances, yes, permeable, yes, very good, permeable membrane. So, now I came to know that you understood the cell wall. Okay, last question about cell wall. Cell wall is present in which cells? I have given you 4 cells there and I have told you that these cells are having the cell wall. Okay, algae, fungi, plants. Very good. Keep it up. Very good, kids. Now the next topic. What is there inside the cell wall? That is the plasma membrane. Plasma membrane. Okay, next topic is plasma membrane. This is always living structure, and this always encloses cytoplasm. Encloses cytoplasm. And it is the only one membrane in animal cells, because animal cells, they don't have the cell wall. So, it is the only one membrane in the cells, particularly animal cells, because they don't contain any cell wall. So, now this plasma membrane, it is living structure, and in cell wall containing organisms, it is formed on the inside of the cell wall. Inside the cell wall. If they have cell wall, then it is formed on the inside of the cell wall. If they don't have cell wall, it is the outermost limiting membrane of this cell. Yes, very good. Now, this plasma membrane structure, let us see the plasma membrane structure. It looks like this. This is the outside of this cell, okay. And this is the inside of this cell where cytoplasm is present. And plasma membrane will be looking like this, okay. This is the lipid bilayer. I hope all of you can see that yellow balls with some 2 lines are there. That is a phospholipid bilayer. They are present on both the sides. Then the red ones, you can see they are the proteins. And these blue chains are there. Some hexagonal molecule like structures are there. They are the carbohydrate chains. And this lipid bilayer, it is made up of phospholipids. And these red ones are the proteins. And this protein, which is having a gap in between, that is called as transport protein. And these blue ones are the carbohydrate chains. So, all of you go through the diagram and try to observe the parts. Okay, after this, I will be asking a small question so that you can answer that. All of you just go through the diagram. Okay, have you seen? Ready for the question? Yes, very good. Now, plasma membrane is made up of which materials, which molecules or which materials are making the plasma membrane? You can see in this structure only the names are given with what they are made up of. Yes, lipids, very good. Carbohydrates, proteins, yes, very good. So, now let us concentrate on this point that I will show you the diagram afterwards. So, plasma membrane is mainly made up of three things are there mainly in this. The first materials with what the plasma membrane is made up of is lipids. That first material is lipid. Then the next materials are proteins. Okay, lipids, proteins, and then carbohydrates. I hope all of you have seen these names in the diagram, right? So plasma membrane is mainly made up of three materials or three molecules. They are lipids, proteins and carbohydrates. So first one, lipids. Lipids are like oils, fats, like that. So in that lipids category also you have a particular name of a lipid which is called as phospholipids. In the lipid category also you have phospholipids. For a lipid molecule, one phosphate group is added. So it is called as phospholipid. That kind of lipids are present in the plasma membrane. Along with that, you know, one more lipid will be there. Some people will be using cholesterol. Okay, cholesterol term is used in Telugu. Okay, like kovu, something like that. Okay, so cholesterol. So that cholesterol is also one kind of lipid. Okay, derived lipid basically, that is also present in the plasma membrane. Particularly in the animal cells, definitely we have the cholesterol also. So lipid components of plasma membrane are two things, phospholipids and cholesterol. So these two lipids will be forming the plasma membrane. And these phospholipids, they will be looking like this structure. They will be having one round head and they will be having the tails like this. Each phospholipid will be having this type of structure. And phospholipids are arranged in the bilayer form. You can see in the diagram, this is one layer, this is the second layer. So it is called as phospholipid bilayer. So phospholipids always they will be forming the bilayer. And among that bilayer, you will be having in some areas the small molecule cholesterol will be present. This will be the cholesterol. So majorly the lipids which are forming the plasma membrane are phospholipids, and minorly very small amount of cholesterol here and there it is present. So that is a basic structure of the plasma membrane because it is mainly made up of phospholipids. Then in the proteins category, we have different types of proteins here. And the type of proteins which are present here, they are integral proteins and peripheral proteins. Integral and peripheral proteins. All of you can see here in the membrane, okay, if I take outside this side and inside this one, here if I write, okay, or here I can write. So towards the inside or across the membrane also we can show that. You can see the previous one in the diagram. All of you can see in this one. This protein is present across the membrane, but some proteins we can write here, some proteins we can write on this side, some proteins we can write across the membrane like this. So all of you see here which proteins we can remove very easily. Like I'll give the numbers 1, 2, 3, 4. Which proteins can be removed so easily? Is it 1, 2, 3, 4? 1, 3 or 2, 4? Which ones we can remove it easily? Anyone? 1, very good. 1 and 3. 1 and 3, very good, very good. So 1 and 3, these two, which are easy for removing, easy of removal, okay? For that, these proteins are called as peripheral proteins because they are present on the sides and they are very easy for the extraction. We can extract them very easily. So these ones are called as peripheral proteins which are present on the side and which are easily removed from the plasma membrane structure. Now all of you can see 2 and 4. 2 and 4, can we remove them easily? We can't remove. So these 2 and 4, these two are called as integral proteins. Integral proteins. Okay, that integral proteins are very difficult to remove and they act as transport. Okay, so they will be transporting certain materials from outside to inside or inside to outside. So based on the function, they have different names like transport proteins, structural proteins, catalytic proteins, like that different names are there for this. So we have basically two types of proteins, peripheral proteins and integral proteins. Peripheral proteins are present on the sides of the membrane. They are not embedded in the membrane and they are very easy to remove. Whereas the proteins which are embedded in the membrane, okay, or across the membrane they are present. They are very difficult to remove, and these proteins are called as integral proteins. So these two will be forming the proteins part, integral proteins and peripheral proteins. Peripheral proteins very easy to remove, integral proteins are very difficult to remove. The next category, carbohydrates. In the carbohydrates, you can see here carbohydrate chains. Can you all see this carbohydrate chains? Blue color ones are given like this. These are the carbohydrate chains. These carbohydrate chains can be added to the phospholipids or they can be added to the proteins. If you add the carbohydrate chain to the lipid, it will form the glycolipid. If you add that protein and lipid, you know proteins are there and lipids are there. If you add to the protein carbohydrate chain, then it will form the glycoprotein. For the lipid, if you add the carbohydrate, it will be forming the glycolipid. So carbohydrate portion always it will be there in the form of glycolipids and glycoproteins. And these glycolipids and glycoproteins are always present on the outer side of the membrane only. Always. They are never present on the inner side. They are present always on the outer side of the membrane. And they act as pathogen recognition centers. Pathogen recognition centers. They can identify the pathogen and they allow the entry of the pathogen, and these are the functions of that glycoproteins and glycolipids. So you can see in this, carbohydrate chain is attached to this protein. Carbohydrate chain is attached to the phospholipid bilayer, only on the outer side. You can see this is the outer side, only blue chains are given on the outer side, never on the inside. So always carbohydrate chains are present on the outer side only, never on the inside, because this will allow the entry of the pathogens through them. They allow the entry of the pathogens through them. Recognition centers. So basically the plasma membrane is made up of three components: lipids, proteins, carbohydrates. In the lipids we have phospholipids and cholesterol. Phospholipid bilayer will be there, and in that cholesterol will also be there in the lipid components. And in the proteins we have two types of proteins: peripheral proteins and integral proteins. And in the carbohydrate portion we have this carbohydrate chain is attached to the protein, so this is called as glycoprotein. Whereas this is attached to the, like this, if you attach the phospholipid, this will form the glycolipid. Okay. Now let us see the last thing, that is functions of plasma membrane. Functions of plasma membrane. It is concerned with cell growth. The first function, it is involved in the cell growth. It is involved in the transport of molecules. You have seen already transport protein is there. Transport it is involved. Then it will form the intercellular junctions. Intercellular junctions. Then it is also involved in endocytosis. Endocytosis means taking in the materials into the cell. Okay. Then it acts as a semi-permeable membrane. Semi-permeable membrane or selectively permeable membrane. Selectively permeable membrane. These are the functions of the plasma membrane, the major functions. So cell growth, it is participating, the plasma membrane, and transport of different molecules like active transport, passive transport, So they are involved in the transport of molecules from inside to outside or outside to inside, like that. Then intercellular junctions, two cells are connected by the junctions, that junctions are formed by the plasma membrane. Then endocytosis, taking in the food materials or the liquid materials into the cell, that is called as endocytosis. Endo means inside, cyto means cell. The process of taking in the materials is called as endocytosis. Semi-permeable or selectively permeable, the word meaning is semi-permeable. It will be allowing all the solvents. I hope you understood the word solvent meaning. Solvent means any liquid. So it allows all the solvents here, but no solutes are allowed to pass through it. It allows all solvent, but it will not allow any of the solutes. Then it is called as semi-permeable membrane. Okay. Selectively permeable membrane means it allows all solvents. No doubt all solvents will be allowed here, but few selected solutes. Few selected solutes only will be allowed to pass through the membrane. So if it allows only the solvents, it is called a semi-permeable membrane. Sometimes it does that function. Selectively permeable membrane means where it allows all the solvents, but it allows only few selected solutes which are required by the cell, or which are not required by the cell, they will be sending it outside. So these are the main functions of the plasma membrane. I hope all of you understood this. So will you be telling me the answer for one question? One small question. What is the outermost limiting membrane in animal cells? Cell wall or plasma membrane? Very good, very good. Cell membrane. Yes, yes. If it is a plant cell, is it plasma membrane or cell wall? Cell wall. Very good. Very good. In the plant cell wall is outermost layer. In the animal cell, very good, cell membrane. So I want all of you to read these topics, okay, cell wall and cell membrane, including the prokaryotic cell, and we'll continue ahead tomorrow with another cell organelles topic. I hope all of you understood. Yes, thank you. Thank you, students. Tomorrow let us meet for the next topic, cell organelles. Thank you.
Hello everyone, how are you all, my lovely kids? Now let us start today's class, everyone. Are you excited for the class? So now, today we will be talking about AIDS. AIDS is a disease which is not curable, beta. So we are going to study the another disease today. Yesterday we have studied cancer, and you know that cancer is the major cause of death. Today we are going to understand about AIDS. Okay, AIDS. Alright, full form of AIDS. Tell me the full form of AIDS. Acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. So let us first of all understand, beta, the meaning of this acquired immunodeficiency syndrome. The meaning of acquired means it is not present by birth. The meaning of acquired means it is not present by birth. It is acquired after birth. Okay, then you will tell me, ma'am, we have heard that if there is a HIV infected mother, the fetus can get the infection. So at that time though it will be by birth, hai na? So now when we say acquired, we mean to say that it is not genetic. We mean to say that it is acquired after birth. It is not a genetic disease. Genetic disease are the diseases which occur by birth because they are present in the genes, beta. Alright, acquired means it is acquired after birth. It occurs after birth. Alright, if it is occurring, if it is coming from mother to fetus also, then also it is acquired. At that time, the fetus is acquiring it from the mother, but not through the genes. Okay, are you understanding this? Are you understanding this point? Coming to the next point, immunodeficiency. Immuno means immune system. Beta, immuno means immune system. And that is why I have already talked about immune system. I have already taught you immune system. Now you know in the immune system, lymphoid organs are there, B cells are there, T cells are there, right? So immune system. Deficiency means there is something which is lacking. There is something in which immune system is deficient, right? So immune system is deficient. Immune system is deficient in what? Immune system is deficient in T helper cells and beta macrophages. These are the two cells, these are the two types of cells in which the virus will enter, okay? The T helper cells and macrophages. You know about macrophages? Everyone knows about macrophages. They are important cells. Macrophages are the monocytes only. Macrophages are the WBCs only. Macrophages do the phagocytosis. They engulf the pathogen. If macrophages will be killed by the virus, then our immune system will become weak. Similarly, Pinky beta, I will tell you everything, the causes, the detection, right? Everything I will tell you, but step by step, right? So macrophages you know, but now you will ask me what is T helper cell? You have told us T cell. Yes, I have told you T cells. Beta, now T cells are of different types. Let me tell you here T cells. T cells are of four types. Helper T cells, helper T cells, killer T cells, killer or cytotoxic T cells, right? Then regulatory or suppressor T cells, regulatory T cells, and then beta we have memory T cells. So we have four types of T cells, everyone. Don't talk among yourselves. Focus on the lecture. Okay. We have four types of T cells, beta. Four types of T cells. But the helper T cells are very, very important cells. Helper T cells will help the B cells also for the production of antibodies. I have told you B cells. I have told you that B cells will produce antibodies. Yes or no? B cells, B lymphocytes, they produce what? They produce beta antibodies. ABs means antibodies. So helper T lymphocytes will help the B lymphocytes in production of the antibodies. Okay, that's why their name is helper T cells. These helper T cells, they help B cells. So, and also the helper T cells also help the other T cells. So helper T cells are very, very important cells. Okay. Now when helper T cells are decreased in the body, whenever there is an AIDS infection, in the AIDS disease, helper T lymphocytes decreases very much. So both the immunities will be compromised. The humoral immunity is provided by antibodies, but helper T lymphocytes are decreased. So then B lymphocytes will not be able to produce antibodies. So antibodies will also be decreased. And the T lymphocytes provide cellular immunity, cell-mediated immunity. So both the types of immunities, antibody-mediated immunity and cell-mediated immunity, both the types of immunities will be compromised and the person will become very weak. Alright. So immune system is deficient in T helper cells and macrophages. T helper cells and macrophages. Now, syndrome means what? Syndrome means with a group of symptoms. That means in this disease, beta, so many symptoms will come, right? Because AIDS is a disease in which other diseases will also occur. I will tell you opportunistic infections. I will tell you what is the meaning of opportunistic infections. Opportunity means whenever you are getting an opportunity, you are utilizing it. Yes or no? For example, now you are getting the opportunity to, attend the STAR program, to get interacted with your teachers before even coming to the campus, you are getting the opportunity. So some of you are utilizing the opportunity very well. Yes, some of you are utilizing the opportunity very well, but some of you are wasting the opportunity, beta. Some of you are only coming here to ask the Instagram IDs of each other. and people are giving also here. Yes, Instagram will not teach you anything, okay? Instagram is just a waste of time for you people. I also use Instagram, okay? I also use Instagram, but for you this time is very, very crucial. This is the class time. Why you are talking about Instagram IDs? This is the class time, beta. Later on, you can ask whenever you get any chance, but utilize this opportunity. So, whenever there is a AIDS infected person, in that AIDS infected person or HIV infected person, opportunistic infections will occur. That means the pathogens will get the opportunity to do the disease. The pathogens will see, okay, this is a AIDS infected person, this is a HIV infected person, so its immune system is very weak. So now we can attack. So pathogens will get the opportunity to, cause the disease. Otherwise, when our immune system is very strong, if our immune system is very strong, our immune system can fight with the pathogens. So pathogens will not be able to cause the disease. But in case of the AIDS patient, in case of the HIV infected person, immune system is weak. When immune system is weak, then the pathogens will get the opportunity to cause the different diseases. There are some pathogens, there are some pathogens which can never cause the disease to us because our immune system is strong. But in a HIV infected person, they are also causing the disease. They are getting the opportunity, and that is why they are not losing that opportunity. They are utilizing that opportunity and they are causing the opportunistic infections. So I hope you are understanding opportunistic infections. What is the meaning of opportunistic infections? Opportunistic infections occurs in a HIV infected person. Beta, HIV is the virus. Movie plus, HIV is the virus. AIDS is the name of the disease. HIV is causing AIDS. Okay everyone, what is the difference between HIV and AIDS? HIV is the virus, beta, and AIDS is the disease. So AIDS is caused by, the next thing is AIDS is caused by, it is caused by HIV. HIV. What is the full form of HIV? Everyone tell me in the chat section. What is the full form of HIV? It is not human immunovirus. Everyone, it is the human immunodeficiency virus. It is the human immunodeficiency virus. That is why it is causing the deficiency of immune system. So it is human immunodeficiency virus, HIV, human immunodeficiency virus. Now I will tell you very interesting things about the HIV. Beta, HIV is a retrovirus. You should remember HIV is a retrovirus. What is the meaning of retrovirus? What is the meaning of retrovirus? This HIV is a retrovirus, beta. Retrovirus. Anybody? Yes. Can anybody tell me what is the meaning of retrovirus? I will tell you. Listen to me. Retrovirus. The retrovirus is it is a family of viruses in which they have RNA genome and they can change this RNA into DNA because they have a special enzyme for that. They have a special enzyme which the enzyme will change the RNA into DNA, beta. Okay, so their genetic material genome is RNA. Retrovirus genome is RNA, beta. RNA genome. In this RNA genome, you should remember there is single-stranded RNA. There is not double-stranded RNA. There is single-stranded RNA. And how many copies of single-stranded RNA? 2 copies, beta. 2 copies of the single-stranded RNA. Along with this RNA genome, they have an enzyme that is, beta, reverse transcriptase. Very, very important enzyme. Without this enzyme, it cannot cause the disease. Without this enzyme, it cannot cause the disease. So, Rian, beta, reverse transcriptase will convert the RNA into DNA. reverse transcriptase enzyme. Reverse transcriptase enzyme. This reverse transcriptase enzyme, what it will do? It converts RNA into DNA. RNA into DNA. RNA into DNA. Do you know the meaning of transcription, translation, replication? Yes, everyone, tell me. Tell me, do you know the meaning of replication, transcription, translation? BTS forever. Ma'am, what is genome? Beta, genome is your genetic material. In every cell of the body there is a nucleus, and in that nucleus there is a genome, genetic material, which can be either DNA or RNA. Retroviruses are those viruses, their genome is RNA, and they also have the reverse transcriptase enzyme. So this enzyme will help them to convert the RNA into DNA. So let me tell you what is the meaning of transcription. Here what is happening? Here reverse transcription is happening. Let me tell you. When anybody will say DNA is converted into DNA, DNA is making its own copies. DNA is making the copies. DNA is converted into DNA. Then it is known as replication. DNA to DNA is replication, beta. This is what? This is replication. This is replication. From DNA, when from DNA RNA is formed, when from DNA RNA is formed, this process is known as beta transcription. Transcription. And from the RNA, when proteins are formed, this process is known as translation. You study all these processes in detail in the botany, okay, in the molecular biology. So that is why this is known as central dogma of molecular biology. Replication is DNA to DNA. Transcription is DNA to RNA. Translation means RNA to protein. Okay, so now what is reverse transcription? Reverse transcription means it is ulta RNA to DNA. If somebody will tell you RNA to DNA, this RNA to DNA, this is beta reverse transcription. Reverse transcription. That is what we are talking about. For this reverse transcription, HIV has the enzyme. HIV, human immunodeficiency virus, has the enzyme. That enzyme is reverse transcriptase. This reverse transcriptase will do reverse transcription. From RNA, it will convert the RNA to DNA. So it can convert its RNA into DNA. Yes or no? Now, have you understood? Have you understood reverse transcription and reverse transcriptase? So reverse transcriptase is helping in the reverse transcription. Yes, everyone, tell me in the chat section. Have you understood RNA to DNA? Now let us continue. I was telling you that HIV is a retrovirus. HIV is a retrovirus. Now I will draw the HIV and I will show you what is present, what is the structure of HIV. Okay, and one more important thing I can tell you, that is also given in NCERT. This HIV is a lentivirus. Lentivirus means it has long incubation period. It has long incubation period. Incubation period. What is incubation period? It is the time taken between the infection and the first symptom. Time of infection, first symptom. This period is known as incubation period. From infection to the first symptom. From infection to the first symptom, that is incubation period. This is the time taken from infection to first symptom, appearance of first symptom. So it has a long incubation period, beta. It can live in the body without showing the symptoms from 5 to 10 years. Long incubation period, 5 to 10 years. Sairaj, beta, it is 5 to 10 years. Okay, 5 to 10 years. Now one more thing. about this virus, it is an enveloped virus. It is an enveloped virus. Enveloped virus means it has an envelope. I will show you in the structure, it has an envelope. So if somebody will ask you HIV is an enveloped virus or it does not have the envelope, yes, it is enveloped virus. If somebody will ask you what other features, then you can tell HIV is a retrovirus, HIV is a lentivirus. Retrovirus means it has RNA genome with reverse transcriptase activity. RNA genome with reverse transcriptase activity. Lentivirus means long incubation period, and enveloped virus, it has an envelope. Okay. Now let us talk about the structure of HIV. If you want to see the structure of HIV, I can make it for you for like this. There is a beta envelope. First of all, I will make an envelope. This is the envelope. In this envelope, some glycoproteins are there. In this envelope, some glycoproteins are there, beta. Some glycoproteins are there. Like this. Some glycoproteins are there in this envelope. This is the envelope, and in this envelope some glycoproteins are there. Glycoproteins are those proteins which have carbohydrate and protein. So these are the glycoproteins. Okay, this is the envelope. In this envelope, what I am making is the glycoproteins. I am making some of the glycoproteins. Okay, glycoproteins. Now, there are two protein coats, beta. Inside you will see two protein coats. You will see two protein coats. So I am making two protein coats here. These are the two protein coats I am making. This is the outer protein coat, and one more protein coat is there inside. This is the inner protein coat. Outer and inner protein coat. Protein coat is there. All right. Then there is inside you will see the two copies of single stranded RNA. This is one single stranded RNA and this is another single stranded RNA. So how many copies, everyone? How many copies of the RNA? How many copies of RNA? Two copies. This is a single stranded RNA or double stranded RNA? Single stranded RNA, right? Then along with this single stranded RNA, there is a reverse transcriptase. So let me make a reverse transcriptase here. This is the reverse transcriptase I have made. Reverse transcriptase. And then two more enzymes are also there. One is this enzyme which is known as protease, and one is another enzyme which is known as integrase. Integrase. So let me label everything now. Let me now label everything. Beta, this is the envelope. This is the envelope. In this envelope, there are glycoproteins. This is glycoprotein 120 and this is glycoprotein 41. These are the two glycoproteins. The 120 and 41 is telling the molecular weight. It is 120 kilodalton and 41 kilodalton. So these two are the glycoproteins. GP stands for glycoproteins. GP stands for glycoproteins. Then there are two protein coats, beta. This is one protein coat. This is another protein coat. Alright. So this protein coat is the outer one and this protein coat is the inner one. Outer protein coat and inner protein coat. Alright. So they can be called as P17 and this is P24. This is also telling you the molecular weight of the protein. Okay. So you can see envelope and then you can see the protein coats. Then you can see the two copies of single stranded RNA. This is the RNA. This is the RNA. This is the single stranded RNA. Then you can see the reverse transcriptase enzyme. What is this? This is reverse transcriptase. This is reverse transcriptase. And then you can see two more enzymes. This can be, we can label it integrase. Very important, integrase. This integrase will help, this integrase helps in integration of viral DNA, integration of viral DNA with host cell DNA, with host cell DNA. I will tell you how it will infect the cell. Okay, I will tell you how it will infect the cell. Now there is another enzyme which you can see here, that is beta, this enzyme is protease. Protease will cleave the protein to convert it into the functional peptide. Okay, protease will cleave the protein to convert it into the functional peptide so that the new virus particle can be formed. You will see this here. Okay, so this is the HIV. Now you have seen this is the HIV beta. HIV. This HIV is going to cause the AIDS. This HIV only is going to cause the AIDS. So when it comes into the body, it infects which cells? It infects two cells beta. One is macrophage and one is T helper cells. Okay, so it will go into the macrophages and then it will start making its copies, so many copies. That is why we say macrophages are the HIV factory. Macrophages. If somebody will ask you the question that which cell acts as the HIV factory? So yes, everyone, tell me in the chat section. HIV factory is which cell? Macrophage. The same it is doing into the T helper cells also. But in NCERT, it is written HIV factory for macrophages beta. So you have to remember macrophages. Macrophages act as the HIV factory. Very important question. Now I will tell you how it is infecting the cell. So in the NCERT, there is a very beautiful diagram which will explain you very well how the HIV is entering into the cell and then how it is making so many virus particles. Okay, see this HIV, this is one virus which will enter into the cell and it will make so many viruses. So let us see how it is making so many viruses beta. So everyone, please focus on the lecture. Everyone, let us see this. This is the NCERT figure. This is the NCERT figure. I hope it is clear to you. Okay. Now you can see the retrovirus. Can you see the retrovirus here beta? Can you see the retrovirus? Everyone tell me. Can you see the retrovirus? Everyone tell me, can you see the retrovirus? Okay. Now this is the animal cell beta. This is the animal cell. This is the animal cell. So I have told you this cell. I have told you that this cell, this animal cell will be either the T helper cell or it can be macrophage. Yes or no everyone? It can be either T helper cell or it can be macrophage. This is the animal cell. Alright everyone? Yes. Now see, this retrovirus is coming and fusing with the animal cell. This is the first step. This is the first step. What we are seeing? We are seeing the mechanism. We are seeing the mechanism, or you can write we are seeing the mode of infection. We are seeing the mode of infection. Mode of infection. So what is the first step? You can see first step is fusion, or you can say it is the attachment. Attachment. You can see that here there is the attachment of virus to the animal cell plasma membrane. Everyone, yes or no? Animal cell plasma membrane. The virus is getting attached. The virus is getting fused. Then what it will, what is going inside the cell? Can you see? This is the virus beta. This is the virus. This is the virus. This is the virus. This is the viral RNA. So what is going inside the cell? What is going inside the cell, animal cell, T helper cell or macrophage? Yes, everyone. Viral RNA, because what is the genome? So it is what is going inside? The genome is going inside, beta. What is going inside? The genome. What is the genome? RNA is the genome. So RNA is going inside the cell. Alright? RNA is going inside the cell. Now this RNA, can you see that this RNA is getting converted into the DNA? Yes, everyone, can you see that this is converting into the DNA? So this is beta viral DNA. Viral DNA. Okay, so let us write. First is fusion or attachment. Second step is insertion of viral RNA into the cytoplasm. This is the cytoplasm, na beta? This is the cytoplasm. Then here there is nucleus. This is the nucleus. This is the nucleus of the host cell. Okay. So in the cytoplasm only, what is happening? The third step is beta reverse transcription. What is happening? Reverse transcription is happening. In this reverse transcription, the viral RNA is getting converted into viral DNA. Why? Because this viral DNA will get attached to the host cell DNA. And RNA cannot get attached with the DNA. DNA only can get attached with the DNA. So that is why first this virus will convert the RNA into DNA, and then the DNA will integrate with the host cell DNA. So yes or no, are you understanding? So the fourth step will be integration of viral DNA with host cell DNA, beta, with host cell DNA. You can see host cell DNA is present in the nucleus. You can see here, this all is DNA inside. So viral DNA is now incorporated into the nucleus, and into the nucleus this is the host cell DNA. Okay. Let us suppose this is the host cell DNA. Let us suppose host cell DNA, and this is viral DNA. Let us suppose this is viral DNA. This is what? This is viral DNA. So they will interact with the help of the integrase enzyme. Integrase enzyme will help, and then it will lead to the formation of this DNA. Beta. Like this you can see. Yes or no, everyone? Are you able to understand now how it is integrating? So this is host cell DNA, this is viral DNA. Viral DNA is getting attached with the host cell DNA. Alright. So now this DNA will replicate. Replicate means many copies of the DNA will be formed. Then those copies of DNA will be converted into RNA. So by using the host cell machinery, the virus is making its viral RNAs. So many viral RNAs will be formed. Okay, so many viral RNAs will be formed. This is going to now replicate. It will replicate. When it will replicate, so many copies of DNA will be formed. Then after replication, transcription will be occurring. Transcription, and then translation. This is the next step. This is the fifth step, beta. This is the fifth step. Replication, transcription, translation. Fifth step. Replication, transcription, translation. That is why I have already told you the meaning of replication, transcription and translation. So, viral DNA will be formed. From DNA to DNA, viral DNA is formed. Then from viral DNA, viral RNA is formed, transcription. And then from viral RNA, viral proteins are also formed. And what is required for the new virus particle? Viral RNA is required and proteins are required. Protein coat is there, na? So proteins are required and viral RNA is required. Yes, everyone, yes. So now the sixth step will be beta assemblage. Assemblage means the assembly, the viral RNA and viral proteins will assemble to make the new virus particle. So now new virus particles will be formed. After assemblage, new virus particles will be formed, beta. New viruses, new virus particles, new viruses are formed. So here you can see, yes or no, retrovirus is coming and attaching first. Then it is inserting the viral RNA into the host cell cytoplasm. Then viral RNA is getting converted into viral DNA by reverse transcription. Then the viral DNA is getting attached with the host cell DNA. Integration. Then host cell DNA is replicating, transcripting, translating. So along with that, viral DNA will be formed, viral RNA will be formed, viral proteins will be formed. So you can see here the new viral RNA, yes or no? This is the new viral RNA. This is with the blue color only. See here, new viral RNA is formed and these are the protein codes. You can see the protein codes here, right? So viral RNA and viral protein will make the new virus particle. This is the new virus particle. New viruses can infect the other cells. They will now enter into the other cells. Yes, everyone. Tell me in the chat section, have you understood how an HIV enters into the cell and how it acts, how it will multiply and how it will make so many viruses? Right, everyone, right. So this was the mode of infection, beta. This was the mode of infection. You will be getting these notes, so don't worry about it. Now let me quickly tell you the transmission, how the HIV transmits from one person to another person. Okay, and so what are the modes of transmission? Is there any treatment possible? No, there is no treatment possible. And is there any diagnostic test is there? Yes, diagnostic test is there. So I am going to quickly talk about three things. One is the mode of transmission. Yes, anybody, can you tell me? Can you help me? Can you help me with the mode of transmission, beta? If there is an infected person, how it will transmit the disease to the another person? Now everyone, mode of transmission. You must have heard that AIDS is a sexually transmitted disease. That means it transmits through the sexual contact with the infected person. The very first mode of transmission is sexual contact with infected person. Sexual contact with infected person. Every time you use infected person, beta, sexual contact with a normal person will not cause AIDS or HIV infection. Sexual contact with infected person, that is why it is a STD, sexually transmitted disease. Then second is blood transfusion with infected person. Blood transfusion with infected person. Okay, with infected person. Then third, using infected needles. Beta, infected is very, very important. Using infected needles, surgical instruments. Okay, surgical instruments, shaving blades, etc. So If they are infected with HIV, then HIV can transmit from one person to another person. Then fourth is from infected mother to fetus through placenta beta, through placenta. This virus can cross placenta and it can go from mother to fetus. So infected mother, everywhere I am using infected. From the normal person it will not transmit, because normal person is not having HIV. Infected person only is having HIV. Alright. So this was all about the mode of transmission. Now let me quickly tell you the diagnosis, detection, diagnosis. Have you heard of a technique which is known as ELISA? ELISA is a technique, diagnostic technique, ELISA. Enzyme linked immunosorbent assay, ELISA. Okay, this is a detection technique beta. We will talk about it in biotechnology. Biotechnology is a different unit in which we talk about ELISA. And the confirmatory test, there is a confirmatory test. After the diagnosis, we will confirm it. So the confirmatory test is beta Western blotting. Remember these two. You can remember these two. What is the diagnostic test? ELISA. What is the diagnostic test? ELISA. What is the confirmatory test? Western blotting. I hope it is clear to everyone. Now, no treatment possible, no treatment, no cure. So, prevention is better than cure. Okay. So, prevention is better than cure. No cure. Death is inevitable. The AIDS person will die for sure. But still we are giving the medicines. So, the medicines we are giving are the medicines which will prolong the life little bit. They will prolong the life little bit, but death has to be, for sure. Death will be occurring for sure. Okay, because so many other diseases will also occur, na? Opportunistic infections will occur, beta. Opportunistic infections. Opportunistic infections will occur. Opportunistic infections. So, if any other disease is occurring, the symptoms of that disease will come. So, group of symptoms will be coming. Okay, diarrhea, pharyngitis, many symptoms will be there depending upon which other pathogens are coming. Movie plus, which other pathogens are coming, depending upon that, the symptoms will change. Okay, beta, ELISA. Okay, let me tell you the full form. It is enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. ELISA. Enzyme linked immunosorbent assay. This is a technique of movie plus. Yes, we cannot stop death, beta. Yes. Alright. Neela, thank you, beta. Thank you. Alright, everyone. So now this was the end of the class. Today also I have taken a little bit more time, but I hope you have understood the important things, the basic things. Alright. I hope you have understood the mode of infection. Right. So you can read it from NCERT once. When you will read it from NCERT, na beta, you will understand each and everything. After this class, if you will read NCERT, if you will read AIDS from NCERT, you will understand each and every line. Bye bye everyone. Take care and God bless you all. And don't forget to hit the like button and subscribe to this channel if you have not. Okay. So just like the session. If you like my lecture, just like the session. Bye bye everyone. See you tomorrow.